if you really want to get that revision absolutely sorted and spot on this Easter, then click the link in my bio and get access to our revision courses over Easter, where the first 4 to 6 hours are totally free of charge. I'm going to run through one of those theories that we find difficult, the stages of muscle contraction, but we're looking at kind of the muscle fibre level. Remember that, and this was in my previous video, calcium ions, when they are released from the sarcoplasmic reticulum due to activity at the neuromuscular junction, then those calcium ions are ultimately responsible for making the muscle contract. What they do is they bind to the protein called troponin, and that ultimately moves a tropomyosin block, which is associated with the troponin, out of the way. So the tropomyosin moves position. We sometimes describe this as a conformational change in the protein structure. When that moves position, it causes actin binding sites on the actin, I should say, to be exposed. And those exposed binding sites are binding sites where the myosin head will bind straight away when they're exposed, forming a cross bridge between the two protein molecules. That connection, that cross bridge, makes the myosin head bend, and as it bends, it releases ADP and inorganic phosphate, and at the same time it moves the actin inwards towards the centre of the sarcomere. Because the ADP and PI, inorganic phosphate, have been released, that means that a fresh ATP molecule can straight away bind in its replacement. So it binds to the myosin head and it causes the release of the actin from the myosin. The myosin and the actin release from each other, i.e. the cross bridge is broken when that new ATP binds. Now the ATP doesn't stay as ATP for long, it gets hydrolyzed dead quick, releasing energy, and what the energy then does is it's used to reset the myosin head to its original position before it bent, which means it's ready to repeat the process and go back to this stage of the cycle. Now this process continues cyclically over and over and over again as long as calcium ions are still present, keeping that block out the way, and there's enough ATP in the local area to drive the process.